Hi, this is Sumin Choi, and I would like to talk about lytic replication, lysogenic replication, the difference between those two, and lastly, lysogenic conversion and examples. So, how do viruses replicate? Because viruses do not have enzymes or organelles for replication, like prokaryotes or eukaryotes, they have to use host material to replicate. And there are two kinds of replications. There are lytic replication and lysogenic replications. So before we talk about lytic and lysogenic replication, there are three differences between lytic and lysogenic. For lytic replication, gene material of the bacteriophage gets translated to protein to make new viruses after insertion. But for lysogenic replication, it goes through prophage stage, where bacteriophage remains active and repressor proteins inhibit prophage gene. For lysogenic replication, gene material is incorporated into the host genome, whereas for lytic replication, genome remains unattached. For lytic replication, they cannot go to lysogenic replication, while lysogenic replication, it can go to lytic replication when prophage gene gets cut off from its host chromosome. So I will talk about each in detail as we go through. But before we go, I just want to talk about the stages that lytic replication go through. And this is here is a little bit of uh, better drawing for that. So first it attaches attachment. So virus tail fiber attached to the host, and tail fibers have attachment protein for its complementary receptor proteins on the host cell. So there's receptor proteins on the host cell, and tail fibers make an attachment to that. So after that, they break into cell wall using lysozyme, and using the sheaths, virus injects its genome to the host cells. So here, if you can see better. And thirdly, is synthesis phase. So depending on the type of virus, translation and transcription have different pathway. Some have single RNA or dsDNA, but at the end of the day, their virus gene make more virus within the cell. So their gene gets translated to proteins like those ones, and they get assembled to a virus at the end. So so assembly. So base tail sheath DNA, capsid, mature head, and tail fibers automatically attach together to form virus. And lysozyme breaks down the cell wall of the host cell, and virus goes out of the host body. So these are the steps. So lysogenic replication is different from lytic replication. So at the first step, it is same as lytic replication. It gets attached, but and excuse me, when prophage gene gets inside of the host cell, the expression of gene is dormant. So here's when I talked about the difference throughout the prophage stage. Bacteriophage remains inactive, and so that's the second step. So sorry. Here's attachment. There's entry, and prophage in chromosome. The third step, which is genetic replication, is incorporated into bacterial DNA. So this DNA gets incorporated into bacterial DNA. And On the fourth step, replication process will incorporate DNA into replicate the second and third generations of bacteria. So they get, so they replicate themselves, and those same DNA are going to be transferred to other types of other bacteria. So last stage, when induction happens, sometimes host chromosome is excises certain types of excises certain DNA from their chromosome, and those excision is going to cause for lysogenic replication to turn into lytic replication, which is going to cause lysosomes to break out from the cell wall, and bacteria is going to get out. So gene material, like from what I said, gene material gets incorporated into the host genome, which is different from lytic to lysogenic, and for lytic lysogenic replication, it can get into lytic replication, but lytic replication cannot go to lysogenic replication. So lysogenic conversion here. Lysogenic conversion is when prophage gene incorporates into bacterial chromosome. Then prophage gene can contain pathogenic genes. The prophage gene gets expressed, and this can convert non-pathogenic bacteria to pathogenic bacteria. And such conversion happens in third and fourth steps of lysogenic replication. So when the DNA of this when the DNA of bacteriophage has a pathogenic gene, and it gets incorporated into host chromosome and they replicate, they are all going to have pathogenic gene, and they're going to express pathogenic proteins which can have devastating effects to human and host animals that bacteria reside at. So I would like to give two examples of lysogenic conversion. So one is vibrochlorate, and it's an article from CTX vibrochlorate exploring a newly recognized type of phage host cell relationship. So as we know, viral cholera causes cholera, and it's one of the major concerns of developing countries. And viral cholera can arise from lysogenic conversion, and one of the ways in which they can do that is CTX phi. This is a type of bacteria that cause lysogenic conversion, and they put their genome into the cholera gene, which those genes are going to express AB5 subunit type of toxin, which is more pathogenic to human than regular ones. And the way they do that is same as described above. They put their gene into the host cells, and they actually have single strand DNA, which is converted to double strand, and they put into chromosomes, and which in DFF sites, and later they produce AB5 subunit toxin. So lastly is streptococci, which causes strep is a bacterial infection disease which can be transmitted by respiratory droplet or contact with an infected person. Virus called theta 149 inserts virulence gene into streptococci. Virulence gene translates to superantigen genes. And streptococci produces superantigens which cause T cells in our body to function in non-effective ways. T cells are part of our immune system are highly activated, which makes streptococci less significant. So what I'm trying to say is they so virus called theta 149 injects its DNA, and their DNA gets incorporated into host DNA, host bacteria, which is streptococci, which causes strep throat. And those genes from 149, they make proteins which cause T cells, which is like defense mechanism. They activate T cells a lot more in our body, and they kind of defend themselves by activating T cells in our body. It's kind of counterintuitive in a way that if you activated the T cells in great amount, the streptococci presence is going to be minimized in certain sense. And I got this from this streptococci super antigen gene among streptococci, and this is from the PubMed. So thank you for listening. I hope you guys have a wonderful day.